Hi, everyone. We'll go ahead and start the webinar right now. My name is Natalia. If you don't already know me, I'm an aquatic invasive species educator with Illinois Indiana Sea Grant, and I also serve as a facilitator for this wonderful group called the Invasive Crayfish Collaborative, or the ICC for short. And for those who are new here, the ICC is basically a regional initiative that's dedicated to improving the collective management of invasive crayfish in the Great Lakes. So our membership is comprised of professionals from across the region who work to advance how we manage and how we communicate about invasive crayfish. So through the ICC, we send out monthly newsletters highlighting recent crayfish literature and news. We host webinars like this one about new research and programs. And we also engage with various audiences and communities that are connected to these pathways of crayfish introductions. So things like the pet trade and the culinary market too. So before we begin, let me give you a few links that could help you connect with us with the ICC. So our main website is invasivecrayfish.org. If you want to be a part of our crayfish community, you can head to invasivecrayfish.org/join, and then there you'll fill out our subscription form, and then you'll be added to the Google group, which is kind of where we send our newsletters, our webinar registration links, and other resources. And then you could find previous webinars at invasivecrayfish.org/webinar archives. And then finally, if you have any questions or any feedback, you can email me at shkladuk@illinois.edu. Okay, so today's webinar is on red swamp crayfish telemetry across seasons and techniques. And I'm sure as many of us here already know, red swamp crayfish are the most widely distributed invasive crayfish today, and their management is an ongoing effort to mitigate their impact on invaded ecosystems. So to support management efforts, researchers at Michigan State University have conducted radio and acoustic telemetry and have monitored red swamp crayfish across long temporal scales to better understand their movement ecology. So here to talk about this study and its findings, we're pleased to have Dr. William Oda from Michigan State University. Thanks for being here, Will. And for those who don't know Dr. Oda, he is a postdoctoral researcher in the Roth Lab in Michigan State University's Department of Fisheries and Wildlife. He is currently studying the impacts of invasive red swamp crayfish in Michigan and the threat of invasive crayfish in the Great Lakes. He completed his dissertation at the University of California, Riverside on the impacts of urbanization and invasion on freshwater communities. So before we let Will take the floor, I just wanted to give some final notes for all of our attendees. If you have any questions during the presentation, you can type them in the chat or in the Q&A box. And once Will is finished with his presentation, we'll all kind of have a discussion together afterwards. Also, this webinar is being recorded and it will be posted on the ICC website and on YouTube with captions so that you can revisit it or share it with your colleagues whenever you'd like. Okay, that is my spiel. I'll let you go ahead, Will. You can share your screen and take it away. You're muted, Will, if you're trying to talk.
There we go. Can everyone hear me now and see my slides?
Yes to both.
All right, fantastic. So as Natalia said, I'm a postdoc at Michigan State University, and I'm here today to talk about the findings of many years of red swamp crayfish telemetry efforts here in Michigan. So invasive species are a huge threat to biodiversity and ecosystem health on the planet, and they are one of the greatest concerns we have for the future of ecological health. Invasives have a huge economic cost, and this is reaching billions of dollars per year as humans connect more ecosystems over time, allowing for more opportunities for species to either invade and establish or to disrupt the natural processes of historic ecological communities. This figure does a great job of sort of showcasing how these flows move across continents and ecoregions. So one of the ways that we can think about the impacts of species is how they move through the environment. So there are various internal and external cues that drive animal movement. Migration is one of the best examples, and I decided to not pick birds for the image, but instead rays, which also do a really cool migration behavior, and is a classical example of how organisms are responding to the cues in their environment. Understanding how these organisms move can inform the management or conservation of species. So, how does this work? Well, invasive species that have, we can utilize their unique life history and movement to target management efforts. So, some examples like here are sea lampreys, where they move into streams for breeding, and we can specifically target them when we know they're moving through these areas. Another example are zebra mussels, and for their movement, we know that one of the great ways that they spread is through human transport on aquatic vehicles, so boats, ships, things like that. So by targeting them as they are moving through this invasion pathway, we can have more effective management efforts to prevent their spread. Same concept for New Zealand mud snails, which can survive their eggs a long time on aquatic equipment, so boots, rods and reels, anything that you take out into the waters where they're present. And by having wash buckets or cleaning stations, we can specifically target them as they move through the environment and are specifically removing this dispersal pathway. So using our knowledge of organisms' movement through the environment can really help the effort level that we need to manage invasive species by targeting them at places where they're very vulnerable. So, as Natalia said, red swamp crayfish are the most widely distributed invasive crayfish in the world. They were first studied, oh, I spelled that wrong, studied in aquaculture, and this was in the early 1900s when they first took off as a widely distributed food resource, and they spread from their native range in the southeastern United States, so along the Gulf Coast, in Louisiana, Alabama, through Texas and northern Mexico. To California and Hawaii as some of the original import locations for aquaculture, to then Africa, Europe, Japan, and China, where their use in aquaculture has its like highest density. So this species was very widely distributed specifically as a food resource or aquaculture product. They then became a topic of study for different behavioral ecology. They're still used today, actually, to study behavioral ecology in many college classes because they're a great model organism and an invertebrate. For any of us who've applied for permits to work with vertebrate organisms, we know why their use is such an advantage over their vertebrate counterparts. But this then led to the study of red swamp crayfish invasion as they began spreading across five of the different continents in the world. And this became one of the more prominent topics of red swamp crayfish study beginning in the 21st century. This research has shown that they are so successful as an invader because they exhibit high ecological plasticity across invasions, which leads to a wide array of ecological impacts. So we know that because of their high ecological plasticity, they are known to alter food webs, water quality, erosion rates, and predation in the systems where they're present. This often comes in the forms of aquatic vegetation loss, changing the benthos, trophic cascades as they remove native species from food webs. And these impacts aren't just ecological, they're also economic. So this species has a negative impact on crops, specifically rice and rice fields where they've been introduced. Aquaculture, if they can get into facilities, they can predate on the actual target species that are trying to be raised, and infrastructure. So they are a burrowing crayfish species, and through their burrowing, they can erode dams, canals, ditches, and change how well these features of the landscape function. And then one of the specific ways that they have been shown to really negative impact biodiversity is through amphibian losses. So they are a huge predator of amphibian eggs and larvae and can really take out whole populations where they are introduced into amphibians' native range and they don't have natural defenses against this type of predation. So moving on to movement, what do we know about red swamp crayfish movement? Unfortunately, not a whole lot. So we know a lot about their impacts in the environment. We know about their use as a food source and how to raise them, but we don't really know how they're moving, and we don't have effective ways at the moment to target them in their environment because of this. So a review in 2021 of decapod telemetry studies found four pieces of work on red swamp crayfish movement. And then internally, we did an additional literature review and found four additional studies. One of the biggest shortcomings is that a lot of these studies are extremely short in time scale or reach. So, an example is one study looked at 1 day of tracking 4 times a year and used that to calculate red swamp crayfish movement speed. The mean time of tracking across these 8 studies was 1 to 27 days. So, this is a single month off, and almost all of them were conducted in the summer. And this is going to really limit our understanding of how they move throughout the year and like what other factors are changing because we're just not getting a very clear picture of what is going on. Another problem is the majority of these studies occurred in Europe thanks to the very valuable efforts of Girardi and Anastasio, who have conducted a lot of the research on red swamp crayfish movement. So, ooh, yeah, okay, I didn't skip a slide by accident. Here's a little table, and we can see that the majority of this work was done in Italy, Portugal, and Spain, with one previous study conducted in Michigan. I'll be sharing some of those results from that study here today with you. I'm very thankful for Maggie and everything that her USGS colleagues did to help make this work possible. But when we look at then the movement metrics and how what we're actually measuring, we see some commonality. So movement per day, movement speed, total distance traveled are frequent measures of what these species do, as well as their sort of behavioral state. So that was a really important thing a couple of studies had looked at in if there is like specific phases of movement. So, what we know from these studies is that red swamp crayfish exhibit a phased movement pattern. So they have stationary and exploratory periods. In this graph here from Maggie's work, you can see stationary and exploratory phases in the two different colors of blue and red. Also, previous work from the European studies has assumed that a home range exists in the species. So this is that there is site fidelity, they're returning to a same general location, and they are sort of true to that area. So 6 of these studies used radio telemetry. This was the most prominent form of crayfish movement analysis that's been conducted. One study used physical markers where they taped colored plastic symbols to the crayfish or glued colored markers to the crayfish. And one study, Maggie's used acoustic telemetry. And some of the metrics that we see is that we have red swamp crayfish movement speeds ranging from 0.6 meters a day to 75 meters a day. This is a huge range. Uh, Something that is moving 1 meter a day is going to have a very different dispersal ability than something that can move up to 75 meters a day. And again, this is constrained by the time scale that we're looking at these things. So if you're getting one measurement across 3 months, if you get unlucky, you could measure a very, very short movement, which is how some of these 0.6 meters mean values showed up versus if we're getting a more fine scale analysis like Maggie's, we're getting this 75 meter a day result. Spatial use also widely varied from 1,000 meters squared over a 30-day period in one study to 20 kilometers squared in 4 days in one study in Spain, where they were looking at red swamp crayfish movement in rice fields that were drying down. Another important finding was that male form 1, so reproductive form red swamp crayfish, had much higher movement than their female or non-reproductive male counterparts. So, red swamp crayfish in Michigan, first established in 2017, and mitigation and control have been ongoing through a variety of partners, led by the Michigan Department of Natural Resources and their adaptive monitoring plan. They have been a really important partner, and they helped to carry out this radio telemetry project at 4 different locations of the invasion in Michigan and 7 ponds across these 4 sites. Acoustic telemetry was also carried out at one of these locations and ponds. And one of the valuable things about this work that we conducted and I'm sharing today is that we have co-occurring radio and acoustic telemetry tracking at the same location. So we have a really great apples to apples comparison of how these methods are going to differ. So here's what I'm going to share with you today when it comes to our results. The first is I'm going to share how we assess the role of seasonality and location or site on red swamp crayfish movement and habitat usage. The second thing we'll talk about is that comparison of radio and acoustic telemetry and how we can use that for red swamp crayfish movement ecology assessment. And lastly, I'm going to put to test the question if red swamp crayfish truly do have a home range. Is their site fidelity? Are they faithful to it? Or are they like a fish species where over time we have many sort of rapid increases in area utilized with no stable asymptote or like uh, flat area of growth in the area they're utilizing within their environment. So, how did we do this? Here you can see one of our crayfish carrying his little telemetry tag that we've glued onto his carapace. I think it's actually like pretty cute, just like they have a little backpack when they're going through our studies. Here are our sites. So we have four different sites here where crayfish were tracked. And let's see, are you able to see a laser pointer when I do this? Nope, I don't think you are because it just changed the slide. But the bottom left one, just to the left of the four, that is where both acoustic and radio telemetry occurred at the same time. And this has all occurred in southeastern Michigan. And here's that specific study site where both acoustic and radio telemetry occurred. It was one of the original invasion points in 2017. It is a stormwater runoff and retention pond with a surface area of about 3,800 meters squared or zero point 0.4 hectares. It only gets to be about 6 feet deep or 2 meters deep in the center of the site. And this is where we placed our acoustic array as well as conducted the radio tracking. So for the radio telemetry, we were able to tag 336 crayfish, 173 were female, 116 form 1 male, and 50 form 2 male. These range in size from 24 to 63 millimeters. This was to reduce the tag burden or the weight of the equipment they were carrying for the tracking to be under 10% of their body weight. This was consistent with other work that was previously done with red swamp crayfish and has been shown to not impact their movement speed. These crayfish were released across 85 different events spanning the 3 years and the 7 different study sites. And we have Aaron to thank for the weekly tracking of these crayfish. This was a huge amount of effort that we put in to locate these across seasons. And if you've ever been in Michigan in the wintertime, you know that winter field work is not the most enjoyable thing. So we're very grateful for all the data Aaron managed to collect despite the very challenging environmental conditions. So we track them year round. We have data from the spring, summer, autumn, and winter to use. We also noted if crayfish were in burrows because we know that red swamp crayfish are semi-terrestrial and can disperse over land. So if a researcher tracked them to a visual burrow or to a new location, this was noted in our data set. So we're able to even see when they were using the terrestrial habitat surrounding the sites. For the acoustic telemetry, 32 different crayfish were tagged with acoustic transmitters, and these were tracked for a mean time of 27 days and a range of 6 to 37 days. The timing of this study ended because there was a chemical treatment applied to the pond that resulted in die-off, so we didn't want to use any data after that point. It wouldn't be very useful for a movement study. This occurred using an acoustic array with 16 different hydrophones and a centralized receiver, so we got really amazing fine-scale data. You can see that in the number of data points. So over 11 million different locations were recorded for these 32 crayfish, and they were then filtered for accuracy to remove acoustic echoes and to make sure that we had the cleanest work possible. And then for this analysis, I filtered this down to be one observation every 10 minutes for the crayfish. So this resulted in a working data set of 93,608 observations. We felt that one per 10-minute sample was a reasonable ecological timescale to apply to crayfish, and it fits some models of statistical independence for these points being calculated. And to test this, we decided to look for the season and site analysis. So look at the effective season, pond surface area or habitat size, and crayfish population density. And we measure that as catches per unit effort, which is not a true measure of population density, and instead a measure of both density and activity in a site as they're seeking food. And we were using baited traps to calculate this catches per unit effort metric. I mostly call it CPUE, but if you see density, I do mean how many we were trapping and removing from the sites. For movement metrics, I'm going to focus on two today. The first is home range or habitat usage, and the second is movement per day. So to look at the movement metrics, to compare radio versus acoustic, I used a t-test to compare the differences. And for sex-based differences, including male reproductive form, we used an ANOVA since we have three groups: female, male form 1 reproductive males, and male form 2 non-reproductive males. For home range, we used the same tests, radio versus acoustic, and a t-test. And then for sex-based differences, again, we used an ANOVA because we have three groups. For the home range, for the base test, we decided to use a mean convex polygon metric of home ranger habitat usage, or sorry, not mean, minimum convex polygon metric. So this takes the smallest possible area to include all the points, and we're using 95%. So we're excluding the 5% of outliers. For our comparison of analytical method, we decided to run a AICC, so Aikiki's index, to see what the best fit model was, and that was an IID anisotropic model. For those of you who don't know exactly what that is, it means that basically each observation is independent and draws from the same distribution, but the distribution itself varies by direction. There's movement of each point. So it's stronger along certain axes. So this predicts a lot using that. So then we used this model type to calculate an automated kernel density estimate of home range to compare with the 95% mean convex polygon home range to see if the analytical method makes a difference in what home range or habitat usage estimates we are finding. And then lastly, to determine if it is truly a home range or if it is just habitat usage, we decided to look at the minimum convex polygons to see if an area increase asymptote is reached in the population. So I have a little figure to help sort of explain what this looks like. So for each crayfish, we use the smallest number of points that we collected that was possible for it to calculate an area. So our minimum size is 5 points, and we can create a polygon like this that encompasses all 5 points. Then for the next point, it increases the area, and we'll look at how big this new red area is compared to the original blue area, so on and so forth to the end of our time series. So for each of these, if there is a true home range, we should see the area increase getting smaller and smaller over time because they are returning to the same sites, showing fidelity or faithfulness to that location. And if there isn't, there should be sudden peaks or spikes where they move away from this area that they were spending the majority of their time in, and then start living in this new location and creating a rapidly expanding area and creating a new plateau of area in our graphs. So what did we find? You can sort of track which section we are here along the bottom through the markers, and we'll start with seasonality and location. So here are some density plots showing how crayfish were using different locations within two of our ponds at one of our sites. Well, for season, we do see that there were significant differences. So movement per day, the graph on the left here shows the distance per day in meters, and then the season along the x-axis. And what we found was that habitat or that movement speed is significantly different by season. And summer, with a mean of 2.4 meters per day, was significantly higher than the winter and the spring. And spring movement was significantly higher than both the winter and the autumn. So we see a range from 2.4 meters in the summer to 1.1 meters in the winter, which was our lowest value. And when we look at mean habitat utilization, which we calculate as the area in meters squared, we found that the summer habitat usage or home range was the highest with 759 meters squared, being higher than spring with a mean of 158 meters squared, and autumn with a mean of 426. Winter habitat utilization did not significantly differ from any other season, but this is likely due to the small sample size of crayfish we were able to successfully track through the winter. We only had 7 where we got 5 different locations for them. It was very difficult to locate them, especially when ponds were iced over. So we think that winter also has much lower habitat utilization. It would make sense because this is a semi-tropical species in its southern part of its range, and we just needed to get more data. Unfortunately, we weren't able to successfully collect that. But so big takeaways is spring and summer movement is higher, and then the habitat usage is highest in the summertime. And when we look at some of the past studies, or think about this in context of past studies, these are when the majority of movement studies are occurring. So we are getting from previous work sort of the peak of activity according to this analysis. Then how did site change all of this? Well, when we look at the site size or how big the ponds were, we can see that there is a significant increase in the movement per day by the pond size. So we had ponds that range from 800 to 3,800 meters squared, and as the pond size grows, the movement per day increased. The same thing for the habitat utilization or home range. We see that there is a much smaller habitat usage on the low end of this range than the high end. So both of these species, or this species, appears to be responding plastically to the physical environment that it is located within these lentic environments. So as the area increases, they're moving more and they're utilizing more of the area within that location. And then how did they respond to catches per unit effort or as a proxy of population density? Well, they decrease their movement. So their movement per day goes down, an inverse relationship to the effective pond size, and the same thing for the area that they're utilizing of the habitat or the home range that they're demonstrating. So we can see we have a CPUE range of zero point 0.5 crayfish per trap we set up to 3.6 crayfish per trap that we were catching. And again, we had two significant relationships. So they are significantly decreasing with higher catches per unit effort, our proxy for population density. And then did our results match up with sex and form? No, they actually didn't. So across our studies, we see that there is not a significant difference in either movement per day or the habitat area used by crayfish of different male forms or sexes. I chose two of the graphs just to sort of see that these are basically flat lines. We don't see a significant difference here. And this was not something we expected to find. We expected to see that these reproductive males were moving more and had larger habitat usage rates than, or habitat usage areas than their non-reproductive male and female counterparts. Next, let's look at the radio versus acoustic. So here you can sort of see some of what is going to happen. On the left, we have our acoustic habitat usage plot. So we can see that there's these really tight clusters of where habitat is being used versus, and then versus the radio on the right, we can see that there's a wider distribution of areas that our analyses were predicting they were using within the location. Well, what did we find? Meters per day was significantly higher, like 20, almost 25 times higher in the movement per day. So this is a mean of 94, whereas for radio, we had a mean of 4.3. So why are we seeing such a huge difference when we know these crayfish are in the same site? There aren't a ton of factors that would explain the difference beyond the methodological differences between radio and acoustic telemetry. Well, here's why. It's because coast length depends on the size of your ruler. So if you measure a coastline with a mile-long ruler, you can't get all those bends and turns. But if you use a 1-foot-long ruler, you're going to get a lot more of those twists in a coastline. Same thing for acoustic and radio telemetry. Because the acoustic telemetry is an automated array and it's collecting data consistently, we were getting pings every 10 seconds per crayfish, and I used a time scale of one location every 10 minutes, we're getting a ton of data, and I've created a hypothetical to showcase how this works. So at hour 0, we release our crayfish and we note their location in a site. Then the acoustic array at hour 4, in this hypothetical, takes another location. Hour 8, it does it again, 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 again. And then the next day, we finally physically go back out to the site and locate a crayfish with radio telemetry. They end up at the exact same point, but we have 7 locations for the acoustic tracked crayfish and only 2 for the radio tracked crayfish. So when we measure the distance that they traveled, we can see in this hypothetical that the acoustic tracked crayfish has a distance of, let's say, 28 meters. Sorry, I have to plug in my laptop, while the radio tracked crayfish only has a distance of 9 meters because we measured in a straight line from point 1 to point 2. So we could be missing a lot of that variation when we are tracking crayfish with radio telemetry infrequently. So for habitat usage or home range, what did this look like? Well, the acoustic mean home range was about 1,550 meters squared, while the mean home range of the radio tagged crayfish was 543 meters squared. So we had a significantly higher home range or habitat usage for the acoustic tagged crayfish than the radio tagged crayfish using a mean convex polygon method. And again, we're going to showcase how the sampling scale makes a big difference here. Ooh, maybe I won't show it yet. I'll show it eventually. When we look at sex and form, what we see is that there again is a significant difference in the radio tagged crayfish at this one location. So male form one were moving more and they had larger home range or habitat usages than the acoustic or than their female and male form two counterparts. Whereas in the acoustic data set, we don't see that there were significant differences in movement rate or in the area they were using. Yes, here's where I'll show it. So home range method comparison. So then we use two different methods, our automated auto-correlated kernel density estimate versus MCP. What we see is that there is a significant difference in the home range area or the habitat usage area for the mean convex polygon method for acoustic tag crayfish, but there is not a significant difference when we use the auto-correlated kernel density estimate. So because for the acoustic minimum convex polygon method, we're using every single point and we're not thinking about how these are correlated through time, we're probably overestimating the home range area, especially because we do know that there is this exploratory phase of red swamp crayfish movement. So because we're getting every single point, even when maybe they're just leaving their home range for just a little bit for reproductive purposes or for feeding, but then returning to it, we're still counting that as part of their home range or habitat usage area in a way that we don't with the autocorrelated estimates. So what we found was that the autocorrelated kernel density is a much better comparison between these two than using the minimum convex polygon method. And here's what this looks like. So on the left, we have a radio tagged crayfish and we have all of its locations within this site. So this was about once every 3 days or so across the month where we were conducting the study. And then on the right, I have condensed all of the acoustic points to be their daily average location. So we can see that these two graphs look pretty similar to one another when we're taking like 1 day at a time of crayfish movement. There seems to be a couple points where they're using it maybe a little bit more, a little bit less. But then if we take into account all of the data for the acoustic telemetry, we get a plot that looks like this. So we can see that there are tons of places where the crayfish are just sort of popping out for a day or two or just moving around for a few hours even across this entire site, but they aren't using it as frequently. So this isn't like the core of their usage in the area. So this is where the auto-correlated kernel density estimate does a better job of finding where the highest frequency of locations are and how they're relating to one another versus the minimum convex polygon method, which basically accounts the entire site being this area that they're using within their environment. And this was supported when we looked at the home range. So using the radio tracking data on the left, we can see how the increase in area changes across every one of those sites. At one site where we had the least data, we don't see a asymptote be reached, but for the majority of others, we do see the plateau, we don't see an increasing plateau effect. And when we look at the mean across all of our sites, we do see that there is a plateau that is reached where there is an increase in area under 5%, which is the value we were hoping to reach to demonstrate a true home range. So what did we find for this? So the radio area increase, we used all of our telemetry data. We tracked crayfish about once every 7 days, and we found a area increase asymptote at about 16 observations, a mean period of 112 days. And this one location does a good job of showing how outlier points could be exploratory movement. So these crayfish were sort of moving in and out of burrows, and they're the area surrounding this pond, but this isn't the core of the habitat they're using. So they appear to be returning to these locations. And this is the site where we didn't have much data, and it still begins to show this preliminary pattern of a core habitat area the crayfish were utilizing. And did we find the same thing for acoustic? Yes, we did. And it was actually much, much faster and easier to reach this. Because of the finer temporal scale, we use much lower thresholds to assess the area increase asymptote. So we called these a 1% area increase and a 0.5% area increase, which we reached after times of 17 and 18 days. So again, we see that there does appear to be site fidelity and a true home range in red swamp crayfish. So, what are the key findings of this work? Well, seasonal shifts in home range size and daily movement underscore the role of seasonality in shaping crayfish behavior. When we weren't looking across seasons, we were going to be missing how their movement changes across the year. So we see that movement appears to peak prior to beginning of recruitment in the fall, and there is a ramp up following the winter and the spring where crayfish are moving around in their environment more. Total crayfish catches also shape movement. This could be due to avoidance of intraspecific competition, which could be a way that helps the species establish as an invasive. So because they are changing their behavior depending on their population density, or their catches per unit effort that we're seeing in our sites, if they're avoiding competing with one another by limiting the space or their amount of movement in the environment, this could give them a competitive advantage. Site size also appears to be a predictor of movement distances, and we're not sure if this is a lentic or lodic or versus lodic location effect. So is this something that changes because of flowing or still waters and the boundaries or constraints that are placed upon movement in these low environments? And if this trend holds, it could help explain some higher movement results in past work where they were studying red swamp crayfish movement in river systems. Sex did not have the same impacts on movement that was found in previous work, and this could be effect again of system type or available space. Because we were doing this work in pond, this was a much more constrained spatial location than a lot of previous studies. So this requires additional insight. We found that movement per day was significantly higher when using acoustic telemetry, and this is likely due to the sampling protocol, and movement speed is highly correlated with sampling effort. So to reconcile previous research, there needs to be some measure to control for how often you are sampling and like how long of a time scale you have between your sampling points, because we know that we are filtering out a lot of the movement distance. They aren't just moving 0.6 meters a day. They are probably moving much, much more than that. It's why they're such a good dispersing species. Home range is also sensitive to method, and it significantly differed using a mean convex polygon approach, which is the most common method of assessing habitat usage, but it did not significantly differ using an automated kernel density estimate approach. So you should be very careful in how you're analyzing this data. We are planning to look at additional metrics to calculate home range, including harmonic mean and dissect tessellations, to see, again, which of these are going to be the most effective for assessing red swamp crayfish movement or crayfish movement generally. Radio telemetry also found differences in home range movement that acoustic telemetry did not reveal. So the sampling effort and the scale at which you do it could either lessen or greaten the differences in movement metrics. And we believe that the finer scale acoustic telemetry data is giving a more accurate representation of the differences. Red swamp crayfish also do appear to have a home range. It reached a stable asymptote using both types of telemetry methods. They just require different timescales to reach that stable asymptote. So we do believe that there is true site fidelity in this species. Here, I wanted to present one last thing. So here you can see the mean convex polygons for the radio and acoustic telemetry. On the left, we have the acoustic. On the right, we have a radio. And we can see again how the acoustic does a good job of capturing the totality of movement. We have a lot more individuals using a wide array of the site versus the radio, where we're seeing a much more constrained location, probably due to the site fidelity, and we're only coming out once a week. We're much more likely to get them in the locations they're using the most often versus catching one of their exploratory phases, especially because crayfish are often nocturnal, or they are moving very early or late in the in the evening probably not the best time for field work. So, how does this tie back to management implications? Well, we were able to demonstrate that red swamp crayfish do change their behavior across seasons, and this can change management efforts effectiveness. If you're trying to do a trapping effort in, let's say, the fall, after they've ramped down their movement, you might not be able to get as many individuals looking around, looking for resources. There is a better time of year for you to be putting in your effort. Location also matters. Local factors alter their movement ecology due to the plasticity the species is able to present in the environment. You can change the effort level or the amount of traps or the spacing of your traps to best capture the species depending on your site-specific factors. When you're analyzing your data, it definitely matters what type of telemetry you're conducted and how you analyze your data. I'm happy to talk to people about some of the lessons we've learned, figuring out what the best approaches are from this work. And lastly, home range is real, or home ranges are real. So one thing to worry about is what are the effects of competitive release and rebound when we're removing certain individuals from a population? Previous trapping literature has shown that red swamp crayfish male form ones, especially larger male form ones, are the most likely to be caught in traps, especially the most commonly used type, G minnow traps. So if we're removing these large males that probably have large territories from the habitat, something is going to move in and take advantage of their home range. They're going to be able to utilize those resources. So we have to think carefully about what the impacts of removal is and the timing of that, because we could be helping the remaining individuals that we don't remove from locations. Thank you all for coming and listening to this talk today. I would like to thank all of our amazing partners, USGS, Michigan DNR, and the Great Lakes Restoration Initiative for helping fund and support this work and conduct the work. All of the amazing technicians who were out there helping to trap and tag crayfish, as well as those who actually went out and collected the telemetry work that was before my time. I also want to thank Morgan Clark for providing all of this wonderful crayfish art that you saw in many of the slides. If you are interested in getting your own crayfish art or work done, feel free to reach out and I can put you in contact with her as well. With that, I would love to answer any questions you all have about red swamp crayfish movement.
Thank you so much, Will. Really great presentation. So now I think we'll transition to our Q&A portion. Okay, we have one now in the chat. How has movement been studied in rivers? How do/might those results differ from the ones presented here?
Yeah, so there is only one study that's been done in a true river. So that was in Portugal, and they were looking at red swamp crayfish movement. They had very high movement rates, so they were seeing the crayfish were moving up and down the river span, and they also found that there was exploratory and stationary movement periods. That work was done by Anastasio. If you don't have access to the work itself and are interested in seeing it, feel free to reach out and I can share that paper with you. And I I think there will be a lot of differences depending on site type. The linearity of those systems versus the constrained habitat is going to really change the dispersal ability and especially the directionality of movement. So I don't want to speculate too much on how this will change the results, but I do think there might be maybe less of a true home range in a flowing system than these still locations because there is so much. I'm not, I guess like disturbance, I guess, that could potentially move a species. Um, it's really hard to predict what the differences are. That is a large physical environment gradient to make predictions across.
Great, thank you. I think, Patrick, you have your hand raised. You can go ahead.
Go
Awesome.
easy on me,
Thank you.
Patrick.
I will, I promise. Good to see you guys, Natalia, Will. Really great presentation. Really interesting work, and it's got my head spinning on or thinking about a few aspects. First, and apologies if you mentioned it, did you have any tagged crayfish leave the ponds entirely?
We did. So yeah, we had a few individuals that literally moved between ponds or they moved into a local irrigation ditch. So they
Hmm.
did demonstrate some overland dispersal in the study. But yeah, that was not a focus, so I did not bring that up. So you didn't miss anything.
Okay, good, good. How far away did you see those move overland? And just curious over
A
top of
couple
your head.
hundred meters.
Okay, so not like
Um,
super far. Yeah.
Not super far. Yeah. So past work, it seems like um what was that? I think it was almost a kilometer of overland dispersal has been shown before in the species.
Okay, that was going to be my next question because, of course, thinking with my prevention hat on, we see folks having boils in their front yard and find crayfish walking down the street and things like that, seeking habitat, I'm assuming. And how far might they go, right? To actually find that, how far can they go is kind of what I would have
Yep.
in mind. So,
I
yeah.
think a kilometer is a safe buffer to think about as like the maximum overland dispersal distance. And that's going to also really depend on time of year. If they find a puddle, they can totally rejuvenate in that total, like really well. They are so resilient.
Yeah, to just avoid desiccation, then just keep the journey going, basically, type thing.
Yep.
Yeah.
Yeah, so I think like if we think about like their invasions in Mediterranean climates, like that 1 kilometer is probably like a maximum because that's where some of that work has been done in Spain and Portugal. So that's probably similar
Hmm.
to like California or the Western US where there are a lot of drier environments. They're probably going to
Sure.
desiccate faster. If they're doing this dispersal in Wisconsin or Michigan, that distance might increase because they can puddle hop, pond hop. There's a lot more just random water around for them to survive in.
Wonderful. Yeah, that's what I was thinking. Okay, cool. Well,
Yeah.
thank you. Yeah, great work. Really, really interesting. And can you please send me the artist info? Definitely interested in those.
Yes, I will.
All right. Thanks, guys.
Thanks, Patrick. All right, we got another question here in the chat. How do crayfish navigate their benthic environments slash home ranges? Are they recognizing landmarks like submerged logs, rock piles, etc., or using chemical cues? Or are they purely constrained by territoriality?
So I do know they use some chemical cues. They do have visual cues that they can utilize as well, but I do not know if they're able to recognize landmarks. And I can't think of someone who has studied memory or recognition. I do know that they know how to return to their burrows, so that could be either a chemical cue or they are remembering a landmark. They are definitely not entirely constrained by by territoriality or within species effects. They probably are using a combination of visual memory and chemical cues to navigate the environment, and specifically, they're using chemical or sensory cues to identify food resources.
There was an extension to that question. If and this person is asking if there are behavior differences in movement depending on if the crayfish population is stocked from aquaculture or in the native ranges of P. clarkii.
We don't know, and I would love to know. So I believe this invasion here in Michigan does come potentially from some like human managed populations. So we're seeing this with a group that probably has undergone some genetic bottlenecks. But that would be a very interesting thing to study, and I wish that I could get permission to put them into some experimental ponds to do that. I would love to answer that question for you, Raymond, but unfortunately, I don't have an answer for you today. And yeah, Carter's comment, it probably is a little bit harder for them to memorize a river landscape, especially after we think about a large seasonal flow event or something that's going to shift that environment. Yeah, I think it probably is more variable than in a pond or lake, but again, I'm just theorizing in my head right now.
Do you guys have any plans to apply these methods to open systems like rivers?
No, because there's none here. But if someone knows a funder who would want to support this work, I am always looking for funding and fun research questions to answer on crayfish. So I would love to be able to answer that in the future.
Great, thank you. If anyone has any other questions, feel free to put them in the Q&A box or in the chat. We still got some time left. Um,
Yeah. If you haven't
I
signed up for crayfish ICC workshops that are coming up in October.
That's a really good point and a great time to announce that. I mean, we've announced it a few times already this year, but yeah, so the Michigan State University and Illinois Indiana Sea Grant are working on organizing some workshops in four or now three Great Lakes states. We already had one a few weeks ago in New York, and the next one is going to be in Illinois in St. Charles on October 1st, I believe. And it's basically a training for natural resource managers and conservation professionals to train them on crayfish monitoring techniques and anatomy and crayfish identification. So it's going to be a full day of workshops, or one full day for the workshop. And then we have another one in Jackson, Michigan on October 2nd. And the last one will be hosted in Lanesboro, Minnesota on October 7th. So I think you can find, I'll actually post the registration link in right now.
Oh,
So we just have that.
thanks, Katie, for posting that.
Amazing. Always one
Yes,
step
you
ahead.
will get to see a lot of us in person if you come. We'll be distributed across these. I'll be at the one in Michigan. If anyone is coming to that and wants to chat more, I'm pretty certain that Natalia will be at the one in Illinois, and you can talk to her there. So if you want to connect more with us, that would be great.
Yeah. Looks like we got another question in the chat. Um,
Yeah. Uh, and for Raymond, no, we didn't. So it seemed like there wasn't a distribution along shorelines or the depth based on size, but there was a preference. So in Maggie's work, when she analyzed the acoustic data, she found that there was a shoreline preference for red swamp crayfish. And I believe, again, my own musings, but I think it might be due to predation pressure. So, if there is a large fish predator that's going to get the crayfish, the center of the pond is probably a little bit riskier for them than the edges. But it does not appear to be a size distribution effect. At least we did not find any size distribution effect within our study populations. I did not specifically try and answer that, Wesley. So the question was, did you notice a change in movement after rain events or other disturbances? I do have gauge data for some of our sites, so that is something I could potentially look at. That actually is a really good thing for me to look at. I, yeah, if you send me an email, let me put it in the chat. And remind me, I will see if I can pull together some type of result for you because that would be really interesting if like maybe a rain event sparks movement. I think the best data set we have for that is the acoustic because of the fine scale. We do only have about 1 point a week for the radio, so that would be hard to assess. But if we had some rain events that happened when we were running the acoustic telemetry, we could potentially see if like the day after or there's a period after a rain event where movement increases or decreases. That would be really cool to be able to see.
are you able to measure any burrowing behaviors by chance with telemetry?
Oh, yes, let me pull it up. So I didn't share it today, but I do have some burrowing results.
Oh, that's
If we have
awesome.
time, I can show it to you all. Well, let me go find the right folder on my computer. Plots, burrow. Okay, so here we can see the percent of time spent in our burrow by site. So when we looked at it by pond size and by CPUE, there were no significant differences in burrow usage or the percent of observations we see in a burrow. But some sites had very different burrow usage frequencies. So at our hotel location where the acoustic telemetry was carried out, there was very low burrow usage. And again, at one of our private sites, there was very low burrow usage. But at all of the golf course locations, we had really high burrow usage. And these are also where we see some large physical impacts. I'm going to pitch Colin Ossenmacher's research. He found that crayfish burrowing was impacted by soil characteristics. So it could be that this burrow usage or time spent in burrows might be a result of like what the composition of soil at the location the crayfish population is at. So his work should be published soon. He just defended his master's thesis, and there might be a recording of it online. Again, you can reach out and I'll see if I can either find some results or preliminary stuff that is able to be shared, or if his master's defense was recorded, I'll see if I can get that recording to share with people who reach out. But yeah, so burrow usage was different by location, but not by some of these other factors like season, site, or population density or CPUE.
So all those golf course sites, they're all within the same golf course?
No, two different golf courses.
Gotcha. Okay.
You know,
Interesting.
but it is a man-made landscape there, so maybe something that we're doing to make it good for golfing is also making it good for crayfish to burrow.
Okay, well, it looks like we don't have any other questions in the chat or in the Q&A box. So I guess we'll just call it. I want to thank everyone again for joining, and thank you, Will, for presenting your research and your findings. It was very, very interesting, and hopefully we'll be able to come back and share some more results in the future. But yeah, thanks again for everyone joining. Hope you guys all have a great weekend.